DNA isn't just found in the nucleus, it's also found in the mitochondria of all organisms and chloroplasts of photosynthetic organisms. It stems from the theory that these organelles were once prokaryotic cells, and that eukaryotic cells came about after one species of prokaryotic cell engulfed another. By studying the mitochondrial DNA, geneticists are able to map out human lineage through the ages. The more identical the mitochondrial DNA between two individuals, the more likely they share a relatively recent maternal ancestor. For example, the theoretical model of monogenesis, that humans moved out of the African continent 100,000 to 200,000 years ago, is consistent with the mitochondrial DNA evidence. The largest accumulation of mutations exists in African ethnic groups. This type of DNA comparison in mitochondria and chloroplasts provide information about the evolutionary path of plants and animals. Also, the genetic variation between members of the same species is a subject of interest to many geneticists. The non-coding regions of DNA between genes is very different between individuals because of the high frequency of mutations that occur in these areas. However, the non-coding regions are still inherited, and an offspring's non-coding region will be a blend of the parent's non-coding regions. The knowledge regarding the genetically divergent similarity in these non-coding regions is used as a way to determine relationships between individuals in paternity cases and DNA fingerprinting. Lines and signs, short and long interspersed elements, are repeated DNA sequences that alternate with lengths of DNA sequences. Lines and signs are intervening sequences of DNA inserted into the non-coding regions of a genome. They're virally implanted genes in the non-coding regions of a genome that are passed down to future generations. The DNA in these regions do not code for proteins but are inherited by the offspring. If two species have the same line or sign located at precisely the same location in their DNA, it can be assumed they share a common ancestor. Recombinant DNA is a branch of genetic engineering that involves combining the DNA of one organism with the DNA of another. The often cited example of how this technology has helped society is that of the mass production of synthetic human insulin for sufferers of type 1 diabetes mellitus. Prior to this, diabetics would use pig or cow insulin, which created allergic reactions in many. But once the human insulin gene was isolated, scientists cut it out of the rest of the genome and inserted it into the genome of an E. coli bacterium. The bacterium took the human DNA as though it was its own. When the bacterium replicated, the insulin gene replicated right along with it. The bacteria's protein-making hardware transcribed and translated the gene to actually produce human insulin. The more bacteria replicated, the more insulin was produced. The insulin was then separated from the bacteria and packaged and sold as synthetic human insulin. Sounds simple enough. Except how do you manage to cut something as microscopically small as a piece of DNA, let alone specific enough to isolate the gene from the rest of the strand, and then insert it into a microscopically small bacterium? It turns out that bacteria make enzymes called restriction endonucleases that cut their DNA strand at specific nucleotide sequences. It's a defense mechanism designed to protect themselves against infection by foreign DNA. There are several types of restriction endonucleases a bacteria may have, different only in the nucleotide sequence where they target their cut. The target sequence of the restriction enzyme EcoR1 is GAATTC. This sequence on one strand has the same sequence running the other way on the opposite strand. The restriction enzyme cuts at both positions, anywhere in fact along the length of the DNA molecule. So you could have several cuts, not just two, as shown here. Cuts made by restriction endonucleases are specific and predictable. Their staggered cut leaves a few unpaired nucleotides on a single strand on each end of the restriction fragment. These short strands of unpaired nucleotides are often referred to as sticky ends. A length of DNA might end up with several restriction fragments. Only one will have the desired gene. The genome of a bacterium is spliced at one place only, with the same restriction endonuclease creating the same nucleotide sequence in its sticky ends. Adding a restriction fragment to a bacterium results in the two DNA molecules combining. The sticky ends of the restriction fragment complements the sticky ends of the spliced bacterium DNA. Finally, the enzyme ligase covalently bonds the strand from one organism to the strand of the bacterium. The result is a recombinant DNA molecule. The bacteria containing the recombinant DNA is then cultured to form colonies in a petri dish. Each colony is tested for the presence of the required protein, say insulin. The colony producing the insulin contains the bacteria having the restriction fragment containing the required gene. That colony of bacteria are separated from the rest and allowed to thrive and replicate on their own, producing insulin as they go. So recombinant DNA is DNA containing genetic material from more than one source. Restriction endonucleases are enzymes that make staggered cuts in DNA at specific nucleotide sequences. The resulting fragments have unpaired nucleotides at the staggered ends, called sticky ends. The same restriction enzymes are used on the other organism's DNA so that the sticky ends are complementary to the sticky ends of the other DNA. Ligase enzymes connect one organism's DNA to the other's DNA after the complementary base pairs of the sticky ends have connected. The organism can now transcribe and translate the gene in the foreign DNA. With DNA fingerprinting, we can subject the genome of a person's cell to a restriction enzyme, and what's left are varying lengths of restriction fragments, that is, pieces of DNA of various lengths. Interestingly, subject another person's cell to the same restriction enzyme and you get pieces of DNA again, but varying in different lengths. It seems that the distance between the restriction sites where the endonuclease cuts varies from one person to another. The variation in lengths of DNA after a restriction endonuclease has finished chopping it up is as unique to a person as his or her fingerprint. The process works like this. Restriction enzymes are added to a sample of DNA. In reality, and for the most reliable results, several restriction endonucleases are used to create multiple restriction fragments. Small amounts of the DNA sample are treated with a dye that glows under ultraviolet light and then added to the wells at one end of the gelatin material. The gel is placed in the solution and electricity applied to it. The negatively charged DNA fragments migrate across the gel toward the positive end. The smaller the fragment, the faster it moves through the gel. The fragments are viewed under ultraviolet light. The DNA fingerprints of an unrelated man or woman are significantly different. But the DNA fingerprint of their offspring will be a combination of the two. Genetics and society. Transgenic technology imports foreign DNA into plants and animals. Using DNA recombination techniques, transgenic biology involves scientists splicing the gene of interest using restriction enzymes and use ligase enzymes to glue it into the genome of a fertilized egg of another organism. The organism is allowed to grow and once born is capable of producing the expression of that gene. The fertilized egg of a goat has a spliced human insulin gene added, so that when the egg is implanted and the embryo grows to term and becomes old enough, insulin will appear in its milk. Pigs are physiologically very similar to humans, and their organs are often candidates for medical transplants. A lifetime of being on immunosuppressants is the cost of this kind of procedure for many patients, mainly because the pig antigens covering the surface of every cell in the organ is a target of the human immune response as it tries to attack the invading body part. In the same way as the goat, splicing out the gene that codes the pig antigen and splicing in a gene that codes for a human antigen improves the outcome of this type of procedure. Splicing in human genes that make antibodies means scientists can infect cows with diseases, have their immune system make human antibodies, and we can harvest these antibodies to make vaccines. The human insulin gene can be added to cows and goats to harvest the human insulin in their milk. A gene from a spider that codes for its silk, a protein, can be transgenically applied to a goat's genome. Again, the silk is secreted in the goat's milk, extracted and spun into fibers that are both lightweight and strong. Golden rice is genetically engineered to include genes from a variety of sources to increase iron and vitamin A content. It's now delivered to developing countries as part of the food aid. Herbicide-resistant plants can transfer their engineered genes to wild plants and fungi, creating superweeds, upsetting the ecological balance. Monsanto developed a strain of canola, itself a product of selective breeding, in Canada, hence its name Canadian oil low acid, that's what canola stands for, that is resistant to Roundup herbicide, also a Monsanto product. Douse your crop with Roundup and you have healthy canola, no weeds or fungi to mess up the crops. All Monsanto wants is every season buy more seeds. However, farmers like to harvest the next crop from seeds from this crop, and farmers like to trade seeds with the other farmers. Monsanto sued a farmer from Saskatchewan when the Monsanto strain turned up in a neighboring farmer's field. The farmer said the wind blew the seeds over or birds dropped the seeds there. Monsanto won the case. The long-term effects on human health are not known. What are the risks of consuming products of transgenic technology? The federal health agencies deem products safe for human consumption, but how long do we need to test the safety of foods before they're considered safe? Advocates claim transgenic technology will improve human health and alleviate world hunger. Opponents object to private corporations having control over global food production. India is rife with poverty and starvation. A company sent over, for free, genetically modified seeds to improve crop yields in a country with barely dust bowls for fields. Interest groups against genetically modified foods convinced the government of India of the potential for harm that it may do to their citizens and the environment. The Indian government wanted to avoid this negative publicity and refused shipment of the seeds. India is still rife with poverty and starvation.